For your next assignment, what you need to do in lab is create what's called a dichotomous key. Okay, so I can't see it very well, but something like this. It's a diagram. I'm going to draw some for you here that breaks down organisms into different categories based on shared characteristics or characteristics that they don't share. The idea is you have to start off with, and I'm going to use these really common organisms to do a real quick little easy one, and then we'll look at some bacteria. I'll do another one for bacteria, some that you have in the assignment, but others typically that you don't, and kind of create one for you. What you're going to be doing is using the assignment that you just did, your last assignment that actually has all the different bacteria across the top, and then characteristics are the results for all the different tests. Okay. Now, some of these tests are going to be helpful in creating the dichotomous key, and others are not. First, we'll just talk about this. So, one thing that you need to have is something among all the organisms in the group that is a shared characteristic or trait. So that's kind of what we start off with. Okay, so the shared characteristic or trait for this group, they're all animals, and they all have these organisms have a complete digestive system. They have different organs, organ systems, etc. Now, what we want to do is pick characteristics that are different, and so we could list all the organisms here, like dog, the lizard, the bird, the fish, the snake, and the sea star. Okay, so that's all these here. I'll write them clear as we go down. And what we want to do is now pick some characteristic or trait that's going to pull out at least one member of the group or more, depending. Now, your choice of what is that characteristic or trait is going to have a big impact on what comes next. So, for example, we could say fur, and we could say, or hair, so we could say dogs have hair, and none of the others have hair. So that would pull the dogs away from the others. And that would be fine. That would be one way of doing it. But there are certain characteristics that are better to look at earlier on. Say for bacteria, we could look at this group and say which ones are catalase positive and which ones are catalase negative. But that's not the main way we usually categorize or classify or talk about bacteria right from the start. Usually one of the first things we talk about would be their gram stain, their cell wall, right? So that would be the first thing. Are they gram positive or gram negative? After that, one of the second things that you would usually wonder about the bacteria are what's its shape or morphology. Is it a cocci, a bacilli, a spirilli, a vibrio? What's the shape of the bacteria? So that would be another characteristic, and a big one right at the very top. That's not something you would usually do later, okay? There's not specific rules on this, and this is why different people could come up with very different keys. Some are more helpful or useful than others, right? And that's because of their choices. Some choices are better choices overall, but you can still get sometimes the same result, but other times you can work yourself into a corner and have it really difficult to find your way out. So in this particular case, we're going to look at a cranium. So for example, so a dog has a cranium, a lizard does, so a skull. All right, a brain, a bird does, a fish does, a snake does, but a sea star does not. Okay, so the sea star, no. Yeah, so this is really like a yes-no characteristic. So now we still have the dog, the lizard, the bird, the snake, and the fish, but the sea star is over here. Next, okay. We're going to say, well, the sea star now would be done. Now, in our assignment in lab with the bacteria, what I'm going to ask you to do is list at least 4 traits as a minimum in your key. And that could be, for example, for bacteria, gram stain, gram positive, gram negative, that's 1. Cocci, bacilli, that's 2. So you really need only 2 others. Grows on mannitol salt, and positive for fermentation of mannitol, produces acid, something like that. Catalase positive or catalase negative. Negative is still a result, so that you can use that. That you wouldn't have to go a lot more. Some of these you may have to have 4, 5, 6 to actually separate them. They'll be so similar. But others you'll be able to do it with a. If you can separate it with say 2 or 3, I'm still going to ask you just to add in a few more traits or characteristics just to help identify the organism. So continue on with our example here. What do we do next? Well, so let's look at other characteristics that are major. Vertebrae. Well, they actually all have vertebrae. All right. How about respiration? So we'll say lungs. Okay, is the next thing. No, the fish does not have lungs. I mean, there's lungfish and so forth, but fish in general have gills. Okay, so now we're going to have the dog, the bird, the lizard, and the snake. So we could look at some other sorts of things. Now we could look at the fur. So we're going to look at hair as the trait. And yes for the dog, pulls out of here, but no for, now we have the bird, lizard, and snake. So we pulled out the sea star, we pulled out the fish, now we pulled out the dog, now we have these guys left, right? So we could look at some other characteristics. You could pick, there's all kinds of things you can choose. Kind of following along these lines, we could do the feathers versus scales. Okay, so feathers will be the next one. Unique characteristic for birds. Yes. For birds, no. For the snake and the lizard. And then you start to get down. Geez, these are both considered to be reptiles. So you're like, well, how am I gonna separate those out? Well, the one thing we do know about lizards is they have appendages, arms and legs. Snakes don't. Right? They have no appendages. They have a cranium and vertebrae, right? But they have no appendicular skeleton typically. So we could just put appendages as the next one. Appendages, and then we would separate them. The lizard, yes. The snake, no. So that's the idea. Your dichotomous key is a diagram, a drawing like this. And at every single point here that we call a node, we're going to have a trait that we can classify as yes, no, or positive, negative. So it either yes has that trait or no doesn't have that trait, or yes is positive for that test, a catalase test, or no negative for the catalase test. That's the idea here. So you have a list of organisms, you have a list of characteristics and traits. Now the thing is, you'll see that some of those characteristics and traits, all the organisms are negative for it, or all the organisms are positive. Some are not as useful. If you have gram-positive organisms, for example, and you have EMB agar, and EMB agar is really only used to grow gram-negative bacteria, you're not going to use EMB in your dichotomous key. It's not for gram-positive bacteria. It wouldn't be there. If you have gram-negative bacteria, then the EMB would be useful. So you're not going to use every single thing on here. I don't expect that at all. In fact, you really shouldn't. If that's what you try to do, just kind of list these in order, your key will be a mess. It won't make any sense at all to anyone, to you or anyone else, okay? So let me go and actually make one for the bacteria really quick for these bacteria so you get a sense of what to do. And some of the things I'll write you may actually be able to use even in your key. But these organisms are ones that I didn't include for you to identify. Usually we would have these actually in the lab, but I reduced the number from like 12 normally down for about 6. Keep this in mind. There's one more step from your lab. So your lab had media, and what were all the types of media, their ingredients, what were the different tests, what were the test results. Then you have bacteria, characteristics. How do they grow with that media? What kind of results do they have for a particular test? That was part 2. Part 3 now is making a dichotomous key. So you're going to make a key on how you would identify organisms based on some of those characteristics or traits. There's one last part part 4, the final part, where I'm going to give you, I'm going to say unknown B. I'll just be something like a letter. And I'm just going to list off a number of characteristics that would have been results of tests. I'm just going to say gram stain, I'm not going to say positive or negative, I'm just going to say pink. And I'll say catalase test, and I'll put bubbles, and that type of thing. So you're going to have to interpret them. What does bubbles mean? Is that a positive test or negative test? What does pink mean? Is that gram positive or gram negative? That's the sort of thing I'm going to give you. Then you're going to have to take that information, look at your dichotomous key, and then figure out who is unknown B. What is the organism? You're going to have to tell me what species it is. So I'll give you an example here. I'm going to pull some of this information. So I have these organisms here: Bacillus subtilis, Bacillus megaterium, a lactobacillus, a corynebacterium, and a staphylococcus. So we're going to start off here with them all being gram positive. Right? They all are. So Bacillus subtilis, Bacillus megaterium, the lactobacillus, and I'll just put S for species, the corynebacteria species, and the staphylococcus species. I'm not going into that much detail right now. So just kind of generic. All gram positive. So what would be the next thing? Shape or morphology, right? So we have, morphology. And so we have bacilli, cocci. So for the cocci, we have the staphylococcus. But that's the only one, okay? All these others, the Bacillus subtilis, the Bacillus megaterium, the lactobacillus species, and the corynebacteria, they're all going to be bacilli. So we're going to say, all right, what do we do next? All right. So you can think, what other characteristics? You can go into different sorts of tests. Now for these, remember, the bacilli belong to a group of bacteria that can form spores. Some bacteria can form spores, the endospores, and some cannot. All right, so we're going to do spore formation. No, and that's the corynebacteria. And the lactobacillus. Whereas yes, endospore formation from Bacillus subtilis and Bacillus megaterium. Okay, so now, okay, good. So now we at least separated the group of 4 into 2. That's the idea. If you can just break a group in half or pull one member out, maybe 2 members out, then that's something good. If you can't pull any out, then you don't put it. You don't just list a characteristic and then just keep all of them together. So if they're all the same, that's not going to be helpful. All right, so now we're going to look at the bacillus. So how do we separate subtilis and megaterium? So what you would have to do is take that list, like your table here, and you would find those 2 organisms, and you would go down the list. You would say, okay, for the this particular, EMB would say we're not going to use that. For McConkey's, we're not going to use that. How about DNase test? Is there a difference between the 2? And so forth. If they're the same, if they're the same, you're going to just keep going down the list until you find something that says they're different. So you can say, oh, here we go. Here's a test that shows that the 2 of them are different. And you can say, if I went through this for bacillus, I would say the Vogue-Prescure test. Okay. Is one of the few things that's going to help us tell the difference between Bacillus subtilis and Bacillus megaterium. So yes, positive test result is going to be the B subtilis, and negative is B megaterium. So far what we would have done is had 5 bacteria here. Pulled out one, the Staphylococcus is a cocci, the others are bacilli. Now, according to my instructions, you would have to add 2 more things. You would say it's gram positive, that's 1. It's a cocci, that's 2. And so we would need to know what are 2 more characteristics of some staphylococcus. I'd give you a species. It wouldn't just be generic like this. It could be aureus or epidermidis, that type of thing. So you just have to add 2 just to give us that. For here, you have it's gram-positive. It's a bacilli. It is a spore former, and it has positive or negative. See, test for the vogue proscure. So that's the 4 traits. Good. You don't have to add anything more. Now we're left with these 2 guys, the corynebacteria and the lactobacillus. So you would have to go through a list, same thing, characteristics. What sort of characteristics do they have? This is the same, this is the same. Here's something that's different. So we'll try the catalase test. And we look at the 2 of them, and they're different for the catalase test. So the lactobacillus is negative result. And the corynebacteria is positive. All right, so that would separate them, and you'd basically be done, all right, at this particular point. If you needed to go further, you can, if I gave you multiple species of corynebacteria, you might do something else, like, the nitrate test, for example, and say, that'll help me tell the difference between 2 different types of corynebacteria. Or for lactobacillus, can they use glucose as well, as lactose, or another sugar as well? So you'll look at other characteristics and see. That's the overall idea of making a dichotomous key. So what you're going to have to do is, from our lab, you have these 7 bacteria that I've given you that were part of your last assignment. All right, so you have the Staph aureus, Staph epidermidis, Micrococcus luteus, and then I put Bacillus megaterium in there as another gram-positive. And then you have 3 others, E. coli, Proteus vulgaris, and Serratia marcescens. What you need to do is start off with all 7 of those as your list across the top. But now they're not all gram-positive. Some are positive, some are negative. So you'll separate them. And you'll essentially have 2 different branches, one for the gram-positives, one for the gram-negatives. Right? And you need to then pick tests that will show you the difference between the 2 of them, and then you'll make a drawing like this. You can do the drawing by hand. You can do the drawing in a computer. There are all different sorts of ways. I'll give you some examples of some really interesting ways of doing it. But you just have to do some drawing like this that shows me your dichotomous key. The thing is, it's not just that this is the next assignment that's graded, is that you will have to use this yourself for the final assignment. And this is what you'll use to help determine your unknown. So if I said, this unknown bacteria is, the gram test showed it was purple. I said it did an endospore stain, and there were no endospores, or I said all cells were pink, no green structures were found. So you have to interpret this. I might say it's a catalase test, produced no bubbles, that sort of thing. So I'm giving you characteristics. You're going to have to go through and say, okay, it's a it's purple, so it's gram-positive. So you throw away the gram-negatives. You say it's a bacilli, so I'm going over here. If I told you that. If I said only pink structures, no green, you would say the green were the spores, the pink is just the cell, so that means it's not a spore former, it's over here. I said the catalase test had no bubbles. That's a negative result. The positive result has bubbles, the oxygen bubbles. So you would say, it's a lactobacillus. And you say, is that a lactobacillus? And then I tell you yes or no, whether you're right or wrong, that sort of thing. So that's what you're going to have to do in the final assignment. So you need to build this in order to be able to solve it. And then that'll be the end of our lab.